Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR PYQs of December 2023 shift 1 or the morning shift. I will be dealing with cell biology, genetics, applied biology, methodology, DNA replication, protein synthesis, biochemistry, and this is part C. This is part 3 of the discussion. If you are new to my channel, please do like, share and subscribe. Coming to the next question. Shown below is the inversion product of the X chromosome of wild type Drosophila. The W plus gene coding for the red eye color is near the telomere. This is the W plus gene. It is near to the telomere. So it is getting expressed. And another gene roughest is close to W plus. So this is the roughest gene that is close to W plus, but towards the centromere. Okay, it is towards the centromere, but both are in the euchromatin region. So W is giving you red color and roughest is giving you smooth color. Why? Because the eyes of roughest mutants have rough appearance. So if roughest is mutated, it will have rough appearance, but here it is not mutated, so it is giving smooth appearance. The inversion bring W plus near the vicinity of highly centromeric heterochromatin and places RST plus a little away from the centromeric heterochromatin. This is the condition after inversion. Now, which of the following phenotypes will never be observed in the Drosophila strain with inversion? Now, if you see the first option, red and smooth eyes. So, okay, you get red eyes over here, smooth eyes over here. Both are not mutated, even being near to the heterochromatin. So, that you can observe in Drosophila strain. White and smooth eyes. So, this is being near to the heterochromatin region. It is not getting expressed, whereas RST plus is getting expressed. So, you get white and smooth eyes. That is also can happen. White and rough eyes, if both are not getting expressed. So white and rough eyes, so because both are near to the heterochromatin region, so white and rough eyes is also possible. But this is getting expressed, no harm, even though near to the heterochromatin. This a little further away from the centromeric heterochromatin, but is getting mutated. That cannot be possible, because if this is not getting expressed, if this is getting expressed even though near to the heterochromatin, and this is getting mutated because it is further away from heterochromatin, that is not possible. So red and rough eyes is the answer. Then next one. A mutant strain of E. coli having defects in one of the DNA repair pathways was identified. To identify the pathways where the mutation occurred, a researcher looks at the following parameters and identified the defect to be in the base excision repair pathway. Based on the changes in which of the above parameters, this conclusion can be drawn. Topoisomerase enzyme activity, that is not required in base excision pathway. So A is not there. AP endonuclease activity, yes, you require in base excision pathway. Expression in MUTL and MUTS, that is required in mismatch repair. So C is not needed. DNA glycosylase activity, yes, you require in base excision pathway, and DNA ligase is also needed in base excision pathway. So B, D, E is the answer. Answer is option 2. So this is the base excision pathway. Here you have got a modified base. The modified base is removed by the DNA glycosylase. This will create an AP site. Then AP endonuclease will act and it will cut the phosphodiester bond. Then what happens is the DNA polymerase will add new nucleotides to the exposed 3' OH region, replacing a section of nucleotides on the damaged strand. Then the nick, this nick, it will be sealed by the DNA ligase and an original intact DNA sequence will be obtained. So that is what is happening in base excision repair. Then next one. A new strain of bacteria was created by introducing an artificial operon to allow bacterial cells to grow in the presence of iron. The Fe plus operon consisted of genes that made the cells capable of tolerating increased iron. So, Fe plus operon consisted of genes that made the cell capable of tolerating increased iron. So, when increased iron is there, they are able to tolerate the increased iron. For efficient functioning of this operon, the following desirable features were considered. Which one of the following can be used to develop this operon? So you need a repressor protein. So repressor, it is a DNA binding protein that prevents the expression of the genes by attaching to the operator. So when there is a decrease in iron content means the repressor will bind to the operator. RNA polymerase will not be able to bind. And iron genes need for tolerating iron will not be synthesized. That is not required also in the case of decreased iron. But in case of increased iron, the iron will bind to the repressor. The iron repressor complex will prevent the binding of the repressor to the operator. So RNA polymerase bind and produce genes for tolerating increased iron. This is somewhat similar like the lac operon. So you need a repressor protein which is inactive when bound to Fe2 plus. Answer is option 3. Then next one. Unknown antigens can be detected or measured by sandwich ELISA, whereas ELISpot assays measure the number of cells capable of secreting particular molecules such as cytokines, which is considered as antigens. Which of the following is true for both the assays? So ELISA, sandwich ELISA, it is a biochemical assay that is used to detect the presence of a protein by directing an antibody against it. And ELISpot, it is a technique that is used to measure cytokine secreting cells. Now the reaction products in sandwich, both the reaction product in both the assays are soluble. No. In sandwich ELISA, the products are soluble, whereas in ELISpot they are insoluble. Reaction products in both the assays are insoluble. That is also not correct. See fourth option. Soluble in sandwich ELISA and insoluble in ELISpot. Answer is option 4. Reaction products in sandwich ELISA are soluble, whereas those in ELISpot assay are insoluble. Now this is the case of sandwich ELISA. So you first coat and plate with capture antibody. Then sample is, this is the target analyte, that is the sample. It is added to the plate and any antigen present is bound to the capture antibody. Then a suitable biotin labeled detector antibody is added to the plate. This will bind to the antigen. Then streptavidin HRP is added that bind to biotin labeled detection antibody. Then the substrate, that is TMB, is added. It will give a colored product. This is the case of sandwich ELISA. Here the capture and detection antibodies bind to different epitopes of the antigen, so that the protein is sandwiched between the detection antibody and capture antibody. That is why it is called as sandwich ELISA. Then in early spots, plate are incubated with capture antibody. Then cells secreting cytokines are added. The cytokines will bind to the capture antibody. Then cells are removed by washing. Then biotin labeled detection antibody is added. See here, biotin labeled detection antibody is added. Biotin labeled detection antibody is added that will bind to the cytokines. Excess unbound detection antibodies are removed by washing. Then streptavidin HRP is added which will bind to the biotin. Then you add the substrate. TMB substrate is added and converted by the HRP to a detectable form. This you can see as spots. And one spot correspond to one single producing cell. That is in the case of ELI spot assay. Then next one. A transgenic line was developed in mustard. The T0 transgenic line was selfed and the insertion of transgene was analyzed in the parental and progeny lines 1 to 5. The tDNA region between the left and the right borders used for transformation is outlined in figure A. These are the ones. The pattern following southern hybridization using probes A and B is represented in figure B. Genomic DNA was digested with EcoR1. The thickness of the band indicates the intensity of hybridization. The following statements were made based on the above observation. So this is probe A, this is probe B. Probe A binds to this region, probe B binds to this region. The developed transgenic line has a single copy of tDNA. Probe A also gives you single band, probe B also gives you single band, so that is correct. The absence of band in progeny 4, okay. Probe A is giving the band whereas probe B is not giving the band is due to incomplete transfer of tDNA during transformation. Even if that is the case, if this much portion has entered, then also probe B will give you the band. If this much portion has entered, then also probe B will give you band. So the absence of band doesn't mean that it is due to incomplete transfer. Progeny 2 is homozygous at the site of insertion. Probe A is not giving 2. Probe B also not giving in 2. So that means there is no DNA insertion. There it is not a transgenic plant. So C is incorrect. Then all selfed progeny of line 5 is expected to show hybridization with both probes A and B. Yes. A5 is having a thicker band in for probe A and probe B. So when you self 5, then it will show hybridization with both probes A and B. So D is correct. So which all are correct? A is correct. D is also correct. So A and D only. Answer is option 2. Then the Cree-Lox system was used to knock out the P53 gene from the mice lung. So P53 gene is, if you knock out P53, then the mice can develop cancer. An immunoblot analysis was carried out as shown below. The following assumptions were made. Choose the correct combination of the above assumption. The LOX-P system did not work since the recombinase was not functional. You cannot say that because it has worked in prostate but not in the lungs. So recombinase did not work cannot be said here. So option A is not correct. The LOX-P sites were introduced under the promoter specific for lungs. No, it was not the promoter specific for lungs. If it was the promoter specific for lungs, then in lungs it would have knocked out the p53 gene. Beta tubulin is a housekeeping gene. It is used as a control. Then tissue specified promoter selected was of the prostate gland. That is correct. That is why it got knocked out in the prostate. The mice died because of being knocked out of p53. The mice did not have an immediate death. p53 knockout means it will develop cancer and then only it will die. So D is also not correct. The knocked out mice developed mutagen-induced tumors in their prostate gland more rapidly than in their lungs. That is correct. Because p53 was knocked out in prostate. So E is correct. C is also correct. So C and E is the answer. Answer is option 3. Then next one. Figure A represents the sites for NCO1 in a binary vector P CSIR 23. The vector is 16,500 base pair in size. Figure B represents the fragments observed when the vector is either digested with NCO1 or double digested with HIN3 and NCO1. The intensity of fluorescence of the 453 base pair fragment is double that of the 516 base pair fragment. Okay, so this one is more intense. So 453 band is more intense than the 516 band. And it is more intense in the case of H plus N, HIND3 plus NCO1 digestion. Okay, you don't get a 453 band with NCO1 digestion alone. Okay. Which of the following statements regarding the numbers and location of HIN3 site is correct? So how many HIN3 sites are there? You have to. So this is the right border, left border of the plasmid. This is the gel picture that you get the bands. Now how did you get these values? So first NCO1 551 N 1633. So minus 1633 minus 551 you will get 1082. So distance between these two NCO markers is 1082. Now 3200 minus 1633 will give you the distance between these two NCO markers. That is 1567. That band is seen here. 1567, 1082. Similarly, for these two also, if you calculate, you will get 2546 and 516. 2546 and 516. Now add up all these, it will come up to 5711. Now subtract 5711 from 16,500, that is the size of your plasmid. So these regions got cut into pieces. What is the remaining portion? That portion will be 10789. 10789. So that is the case with the digestion with NCO1. Now NCO1 plus HIN3 digestion. Now if you see the gel picture, you will see that 1082 and 516 they are intact. These two bands came from NCO1 digestion. So there is no HIN3 site there in these regions. That is 1082 and 516. There is no HIN3 region over there. Okay. In other places, that is 2093, 10336, 1185, 453, 382, HIN3 digestion has given these bands. So first we will see this one, 10336. 10336 means somewhere here HIN3 should be there. So subtract 10789 minus 10336, you will get 453 band. So the 453 band is here. That puzzle is solved. So one HIN3 site belongs here. Next, if you see for 2093, 2093 means it will come over somewhere here. So 2546. 2546 minus 2093, you will get another 453 band. That is why the 453 band is more intense in NCO1 HIN3 digestion. 2 bands of 453 are obtained. So that is also solved. Then next comes 1185. 1185 will be somewhere in this region. So 1567 minus 1185, you will get 382. So 382 band and 1185 band. So all the bands have been solved. So HIN3 marker is present over here also. So how many sites for HIN3? 1, 2 and 3. So which of the following statements regarding the numbers and location of HIN3 site is correct? It is, there are 3 sites of HIN3, one of which is located in the 1.6 kb fragment generated by NCO1 digestion. That is correct. So the answer here is option 2. Then next one. Following are marker enzymes that would be used to identify correct subcellular fractions. Lactate dehydrogenase. Lactate dehydrogenase is found in the cytosol A3. Okay, option 3 itself is the answer. A3. Acid phosphatase is your lysosome B1. Glucose 6-phosphatase is microsomes C2. And catalase is found in the peroxisome D4. So this was an easy match the following. So answer is option 3. Now next one. The following statements are made regarding the principle and uses of positron emission tomography. So PET principle, I will tell you, it is a radioisotope is attached to the drug. A radioisotope is attached to a drug and is injected into the body, and this is called as the tracer. Now this radioisotope, it undergoes beta plus decay and a positron is emitted. This positron, it will interact with an ordinary electron and 2 particles annihilate, and 2 gamma rays are emitted in opposite directions. These gamma rays are detected by 2 gamma cameras to form a 3-dimensional image. So first statement. The PET scanner is a positron ray detector. Just now I told you it is a gamma ray detector. Gamma rays are detected by 2 gamma cameras to form a 3-dimensional image. So A is not correct. They have asked for correct statement. The PET scanner cannot determine the location of collision between positron and electron in the brain. It can detect. That is the principle behind the PET. So that is also not correct. The typical PET activation studies can measure the absolute metabolic activity of the brain, not absolute relative. So C is also not correct. In PET, a radioactive isotope is introduced in the blood as tracer that rapidly decays by emitting a positron from the atomic nuclei. That is the principle. While telling the principle of PET, I told you that. So D is correct. So the answer here is only D. 4 is the answer. Then next one. In DNA footprinting, the DNA is labeled by random priming so that the entire DNA is labeled. No, only the 5' end is labeled, not the entire DNA is labeled. So A is not correct. They have asked for a combination of all correct statements. The DNA is end labeled so that the bands get organized from higher to lower size after electrophoresis and autoradiography. That is correct. A sequencing polyacrylamide gel is used to resolve all fragments distinctly. That is also correct. Fragments are smaller in size, so polyacrylamide gel is used, agarose is not used. A high concentration of agarose gel is used to resolve the finer bands. That is not correct. Which of the following option has the combination of all correct statements? B and C. Answer is option 3. This is the DNA footprinting. Here, these are the DNA fragments. They are labeled at the 5' ends. This is 5' end labeling. Then you take one set as control and to other set you add your protein of interest which will bind to the DNA fragments. So here you get only DNA fragments but without the protein of interest bound to DNA. Now you treat the control and the sample with the DNase 1. Then you run the page gel, polyacrylamide gel. For the control ones you will get the digested fragments, whereas for the treated sample you will get to see a blank space over here where no bands are observed. That is called as the footprint. Why did you get no bands over here? Because the protein got attached to the DNA and it prevented the DNase 1 from digesting the DNA in that region. That is why you got a footprint there. This is called as DNA footprinting and it is used for studying DNA protein binding studies. Then next one. An oligopeptide is subjected to amino acid analysis and found to have the following composition. The following statements represents outline list the results obtained after analysis. The above oligopeptide is subjected to N-terminal Edman degradation revealing tyrosine residue. So in all the option you can see tyrosine as the N-terminal one. Trypsin treatment did not affect the peptide. That means trypsin cleaves at the C-terminal of arginine or lysine. Here there is no lysine but arginine is present. Arginine is present at the end. Okay, 1 okay. In 2 also arginine is present at the end. So 2 is also correct. Okay. But 4 also arginine is present at the end. So that is all. That also can be taken into consideration. But 3 we may not have to see. Cyanogen bromide treatment yielded a dipeptide, tetrapeptide and free arginine. So cyanogen bromide, it cleaves the carboxyl terminal of methionine residues. So first one if you see it will cut here 1 and then 2. So you will get a tetrapeptide, a dipeptide and a free arginine. 1 is correct. But if you see over here it will cut here, then you will get a free methionine but not free arginine. So that is wrong. Fourth one also you will get a cut over here and a cut over here. So again a free methionine will be released. So that is also not correct. So first option is the correct answer. Chymotrypsin treatment yielded dipeptide and a tetrapeptide. The composition of tetrapeptide was valine, arginine and methionine. That is, it got cut over the carboxy terminal of phenylalanine. That is chymotrypsin, it will cut the carboxyl end of aromatic amino acids. So you get a dipeptide and a tetrapeptide. So the correct answer is option 1. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.